It's my pleasure to welcome everyone to the celebration of the recipients of the 2025 NAASC Dissemination of Arabidopsis Knowledge Awards. I'm Gloria Moudet. I'm a faculty member at Wake Forest University and president of NAASC this year. And so a few housekeeping points before we get started. If you want to see the captions, you can turn them on in the lower bottom of your Zoom panel under the More button. This event will be recorded and posted on the NAASC website and mailed to anyone who registered. You can go ahead and keep yourself muted and your camera off during the presentations. And we will have questions for all the speakers at the end that you can ask either by raising your hand or by using the chat. So I wanted to begin by giving you a little bit of background. I think most of you know a fair bit about NAASC. We are a group of elected faculty and early career scientists. We began in 1992 and we're part of the Multinational Arabidopsis Steering Committee. And as you know, we organize the RCAR meetings when they're held in the North America, which will include the next one in North America will be 2027. And so there are two other awards ceremonies that were recorded, or awards webinars that were recorded earlier this year, which are on the website that you can go to. So I want to tell you a little bit more about the NAASC Dissemination of Arabidopsis Knowledge Award. So it really recognizes and honors individuals that demonstrate extraordinary commitment to disseminating knowledge in the Arabidopsis community and beyond. So they were, all these individuals were nominated and selected by a group of NAASC faculty members for their long-term commitment for supporting plant biology through innovative teaching, excellent in outreach, communication, or other ways that benefit Arabidopsis researchers. And so we encourage you in the future to nominate other folks for the awards. We have openings for the awards for next year. And so we'll hear from these individuals about what they've done and their positive impacts on the Arabidopsis community and beyond. And so hopefully all of you are already members of the Arabidopsis community, but we encourage you to join if you have not already done so, and to participate in all the things that NAASC does, and especially to nominate other individuals. We all work so hard as faculty members, and it's really amazing when somebody says, hey, these folks have done a great job. And so think about that over the holidays and people who've made an impact on you and how you can nominate them for awards. We also encourage you to join the Arabidopsis community. Small membership fees help us do things that are important to our community, including organize the ICAR meeting. So now I'd like to get started, and I'm going to turn this over to Joanna, who's our executive director and who will introduce our first awardee. Thanks, Joanna.
Hello, I am happy to introduce our first awardee, who is Nagel Illouz-Eliaz. She is the 2025 Early Career Scholar recipient of the Dissemination of Arabidopsis Knowledge Award. Nagel is currently a postdoc in Joe Ecker's lab at the Salk Institute, and she previously did her studies at the Hebrew University of Jerusalem, where her PhD focused on gibberellin signaling in tomato. Her interest in drought stress is what led her to her current work to improve plant drought tolerance. While she has published in many great journals, such as the Plant Cell, Plant Phys, and Nature Communications, among others, this award recognizes her excellence in outreach, communication, and teaching, as well as mentoring. And I think my first in-person meeting from Nagel was opening day of ICAR 2024 in San Diego, where I ran into Nagel, who was excited to tell me that the children's book she had authored had just been released. I'm sure she'll tell us about it, but Arby, the model plant, is an illustrated book aimed primarily at children and is designed to introduce basic plant science and Arabidopsis research concepts to children in a captivating and accessible way. Nagel has also shared her knowledge through various podcasts, on topics such as the importance of plant research to a general audience and her career in plant biology. One of her supporters wrote that Nagel has consistently demonstrated exceptional dedication to communicating complex scientific concepts to a diverse audience and effectively disseminates Arabidopsis research globally. Others spoke about her outreach efforts and engagement at conferences and symposia, as well as her mentorship and teaching of students. For all her efforts, I congratulate Natanella on her award and would like to turn it over to her so she can tell us herself about everything she has been working on.
Thank you so much, Joanna, for this kind introduction. I'm very grateful for being a recipient of this award. And I think just having an award for dissemination of Arabidopsis knowledge says a lot about this community and the people behind it. And so thank you for this recognition. As I thought of what could I bring to you today in this few slides. No science from me today at all, but I started with a question, as often us scientists do, and I asked myself, why did I write this book about Arabidopsis? And why do I go into classes and talk to children about it? So I really focus on the dissemination of Arabidopsis knowledge outside of our community. And when I thought about this question, I came up with this idea that we have two parallel paths in life, at least for me. I have my personal, professional development life, my academic career, but just growing up, for me, it was joining the army, hiking with my friends, getting married, having children, degrees, friends, etc. Many, many things. The race of life, as we call it. And there's also a parallel path while we're running in this race to becoming the adults that we are. And for me, as I looked into that process, Arby, the Model Plant, this children's book, is just a part of things that I've done before, which include mentorship initiatives mostly for advancing women in science and leading projects in youth hostels that were related mostly to connecting through nature and other things. So this book started with the idea of having every child know or have a different model of what is a scientist other than just a Nobel laureate, and know about plant biology and that it could be a profession. And thinking of why I always had these parallel paths in life, doing something other than just this race, I think stems from two things. One is my parents, as you see here with my older daughter, which is now 12, but this is a while ago. They had their own very intensive career, but they didn't really talk about it much, but they always were doing something else, and something that would be related to giving back to the society we lived in, and also growing up in a kibbutz, which is a way of communal living where the society or the community is in the center and not the individual. I think makes it natural for me to have these additional activities alongside my personal professional development. And so about the book, just a few more slides. This is taking you behind the scenes of the process of writing and illustrating the book. For me, it was very important in theory to have every child read this book. So every child, I don't have any scientists in my family. I didn't have scientists. I don't have scientists today. And I didn't have this thought that I could become a scientist. It just wasn't on my radar at all. So the idea here was, through a compelling story with beautiful illustrations to kind of capture children to read it, and through reading just a nice, easy story, to learn what we do as scientists and try to get them excited about what I really love, which is plant biology and how Arabidopsis helps us study that. So after a lot of work, the book was finally published, and I think it came out the week of ICAR in San Diego by chance, which was amazing. And I started getting pictures of children from around the world reading it, and that was most excited for me. Then I still do go into classes. I was in class last week, most recently, and I go into classes around San Diego and talk to the children about what it means to be a scientist and what do we do in simple words, and then a bit about stress, which is something I'm very interested in. What is stress for them? How do they deal with stress? And then how do plants deal with stress? What is a model organism? Why is Arabidopsis an amazing model organism for us? And I am always surprised of how engaged the children are. It's really a discussion, not like a lecture. We discuss things and they are really interested, even if at the beginning they're a bit hesitant and we only have like the three same people raising their hands. At the end of the class, almost all of them, of course not all of them, but a lot of them would then engage and discuss. And it's always surprising how interested they are in this topic. There's always a reward part for giving back. I think we never expected, but such as this award, which I'm very grateful for, but also feedback from the children that I meet in those classes, which I sometimes get to hear, and that's very exciting for me. With that, I'll hand it back to Joanna or Gloria. I want to thank everybody for being here and encourage you to find your own parallel path.
Very nice. Thank you very much. Caroline will do our next introduction.
Super. It's a real pleasure to introduce my colleague and friend, Professor Don Nagel at University of California, Riverside. Don is a really powerful scientist whose research addresses how the circadian clock influences plants' responses to changes in environments. And she's been particularly interested recently in how heat stress responses are modulated during different times of the day. This data, which of course generates copious transcriptome and translatome data, does have the obvious potential to increase climate resiliency of crop plants. But it came with a problem, right? This copious data needed not only to be shared with the public, but also disseminated more broadly to the Arabidopsis community. And so Dawn and her team really went out of their way to prioritize sharing this knowledge by generating a user-friendly web app, which she'll tell you more about. Which supports the sophisticated analysis of circadian data sets in multiple plants. Of course, Dawn has also been really having a powerful role in disseminating Arabidopsis knowledge through mentoring and training the next generation. I will give you one example, which is that Dawn single-handedly initiated and ran her very own summer research program for 13 transfer students from underrepresented groups. And this was across multiple years. Now more than half of them have gone on to a career in STEM or to graduate school. So excitingly, she's now the director of our RAU program, which is going to broaden her influence even more. And finally, she's been playing important leadership roles in ASPB and NAASC to alter and disseminate the Arabidopsis research more generally. So everyone, please welcome Dawn Nagel.
Thank you. Thank you so much, Caroline. Okay, so yes, I want to thank you again, Caroline, for that very kind introduction. I would also like to thank NAASC, right, the selection committee. It's truly humbling to receive such an award for, the work that we've been doing over the past couple of years or more. So I'm going to try, since we don't have a lot of time, and it's a very difficult talk to give because it's not a research talk, right? So I'll just try to highlight some of the ways in which we think about how, we can share our information to the community. So as Caroline mentioned, our lab primarily studies how plants deal with temperature stress or stress in general. And we take this through the lens of the circadian clock and time of day. We generally use a lot of omics approaches across looking at multiple levels of gene regulation. So this allows us to generate a large amounts of data sets and information that we need to not just synthesize, but also make it available again to the community. And that's really an important part of what we do. A lot of our work has been pioneered or started in Arabidopsis, but we're slowly branching out into other plant species as well. And I've been very fortunate to have a lot of my work funded by either through my NSF Career Award, more recently by an NIH MIRA Award, and I've gotten a lot of support from the university through seed funding that have really helped us to continue to do this research. So over the years, we've published several papers. I'm not going to go into details of these, of course, but this is a way in which we can share our information through the research. One of the things that we found was while doing a lot of these large-scale analysis was that we really wanted to be able to integrate publicly available data. And this allowed us, of course, to make better conclusions from our data, better observations. But it was really difficult to really get access to published data, right? So a lot of times you can get information through, a series of supplemental Excel files or data, and a lot of these type of data lacked information, right? That was really vital for us because we're really interested in time of day information. So we were pretty frustrated at some point, and we wanted to make sure that when we publish our work, that people can easily access our data, right? And it is user friendly. So with the help of a really brilliant postdoc in the lab, Tutwan Banat, who was not just a great bench scientist, he was also a great computational biologist. We decided to develop these web applications. So every time we did a project or published a paper, we wanted to also make a web application where people can actually use our data and also provide the way in which we do our analysis. So all of our publications also have GitHub repositories where we share the codes and the files that we use. So this was one of the ways in which we can contribute to the community. And I'm pretty sure that Nick can relate to this because we've actually used a lot of the ePlant resources, right? It's a great resource. If it's there, people will use it, right? And the same for Diurnal, which was developed by Todd Mockler, which was also instrumental and continues to be instrumental for the circadian community, right? So having a way to actually visualize data, I feel like is way more powerful for people to be able to continue to do their research. So one of the things that I'll touch on very briefly is this web application that was mentioned by Carolyn, which is a way for us to be able to visualize circadian gene expression. Are there heat responses depending on the time of day, the transcriptome or translatome level? Can you look at the recovery dynamics? What happens after you remove that stress? Are your gene of interest regulated by the circadian clock? So we've integrated chip sequencing data from that's been published. And also whether or not your timing of gene expression, so is your gene primarily expressed in the morning, and is that enriched compared to everything else in the genome, right? And you can statistically determine this using this database. One of the powers of this database, which I felt was needed, is to be able to look at multiple genes, right? So sometimes you work on genes that belongs to a small gene family or larger gene families. Can you look at all of those genes at the same time and get an idea very quickly of whether or not your genes are responding to heat stress in a time of day dependent manner? One of the things that we're doing now is, when we developed this, we did it in Arabidopsis because that's where the work was. But now we've been integrating many other plant species as well. We've gotten to the point where we can integrate, as you can see, multiple circadian oscillations from different plants. But we're still trying to get the heat stress responses in there because not every plant species have that kind of information and the type of resolution for us to do that. So we're working on getting that done in the coming years. in addition to just research, this also allowed us to develop projects for students, so we can connect with community college transfer students, allow them to do some research in our lab over 10 weeks. They get curious, they're interested about plant sciences. They learn and many additional types of ways in which you can share your information, whether it's through poster presentations or oral presentations as well. And many of them have actually been so excited about plant research and science that they've gone on themselves to either, continue to be in careers related to sciences or academic careers as well. And they're now training the next generation by mentoring their undergraduate students, which is really rewarding to see for many of these students. So I'll stop there. I mean, there's many more I can say, and I can answer questions during the Q&A, but I would like to thank the members of my lab, current and past members, who've really helped us to develop these resources so that we can share it with our community, collaborators, and the funding source for this as well. And again, really want to give a heartfelt thank you to Joanna and to the NAASC community for recognizing the efforts that we do in terms of research and also sharing information, meeting organizers, all of the nominators that share their kind words, letters of support, and also for the community for using our resources and continuing to use our resources and and citing our work, of course. Yeah.
Our next introducer is Siobhan.
Hi, I am very pleased to be here today to introduce Nick Provart, who is one of the recipients of this award for the public dissemination of Arabidopsis knowledge. Nick has been actively involved in the Arabidopsis community for really over the last 2 decades, and he is well known for providing genome scale data in an openly accessible and intuitive format. He's also been a leader of the multinational Arabidopsis community for many, many years. And most importantly, he's enabled global access to Arabidopsis data without any financial or education-based barriers. So to everybody, no matter where they're at and what they have. Nick has also been a leader in education. He has developed multiple online courses, which he continuously updates. These utilize both Arabidopsis methodologies as well as data. And he's also been a long-term instructor and former organizer of the Cold Spring Harbor Frontiers and Techniques in Plant Sciences course. Finally, he is a respected colleague to many of us in the community, helping us determine how to best visualize and provide our data. And he's also actively involved in the promotion of many younger scientists within the community. So thank you, Nick, and congratulations.
Thanks so much, Siobhan, for that really nice introduction. All right, so I'm going to tell you a little bit about plants and computers and people. And at the risk of giving away how late career I really am, I'm going to show you my first computer program from 1981. And this was entered using punch cards. So that really is quite ancient. But I've always been interested in plants and computers, and I spent a summer in 1982 detasseling corn for hybrid corn production in southwestern Ontario near Chatham, which was backbreaking work. But I saved up my pennies, and I bought this beautiful machine here, Commodore VIC-20, which had 3.5 kilobytes of RAM and ran at a clock speed of 1 megahertz, and cost me $500 back then, which is $1,475 in today's funding. I then, fast forward a few years, I went on to do a PhD in Berlin with Lothar Wilmitzer at the Institut für Genbiologische Forschung, just sort of a standard molecular biology PhD. This institute went on to become the Max Planck Institute for Molecular Plant Physiology. And I was interested in guard cells of droughted plants, potato plants, actually. And we did some transcriptional profiling of what's going on in guard cells using some guard cell specific RNA that I was able to isolate and develop a method to isolate. You can see these things. These are called northern blots, for those of you who don't know. They show some changes in gene expression in genes related to organic osmotica and inorganic osmotica production in guard cells in response to drought. And so we sort of, made some models. Then after that, I went on to help co-found a plant biotech company called Plant Tech, and the internet was just coming into existence back then. And I actually wanted to create a really nice web accessible database for some GMO starch that the company was trying to develop. And it's pretty amazing what you could achieve with JavaScript in those days. And this is kind of a screenshot of Starch Bank. After that, I moved to Syngenta and I became a staff scientist at an institute that was initially ran by Novartis and then became part of Syngenta, the Torrey Mesa Research Institute. And they had kind of the first microarray DB that was this interesting model where they kind of did the experiments for people. And they had an in-house database, but these data were not made publicly available. But we were able to mine them together with Joan Chen, my co-first author on this, to identify transcription factor functionality under different stresses. And based on that work, I was able to get a position at the University of Toronto starting in 2002. And here we created the first one of the first open access plant expression databases called, at that time, the Botany Array Resource. With my first graduate student, and Siobhan actually is a co-author on that paper. It wasn't really until 2005, though, that kind of took off with the invention of this tool called the EFP browser. And here I did a screen grab of the email to the Arabidopsis news group in December 2025, telling people about our electronic fluorescent protein tool is what we called it. And this was actually inspired by Sean Cutler's GFP cDNA fusion paper and put together by a couple of very talented undergrad students, Debbie Winter and Ben Vinegar. And that really sort of was very popular. And I remember in 2006 in Madison, Wisconsin at the ICAR there, seeing screen grabs of the EFP browser images in posters. And I was really quite moved by that. So the bar has been online for many, many years. And we have about lots of genomic data, not only from Arabidopsis, but from other species, at about 5 million page views per month. It's very popular around the world. I think every single country used it last year, which is very gratifying. And in 2023, we became a global core biodata resource, just one of 52 worldwide. So here's old and new, the VIC-20, 3.5 KB of RAM, and here's the bar, which has 256 gigabytes of RAM, basically 75 million times more RAM, and runs at about 1 million times faster. And it only costs 7 times more than the VIC-20 did, actually, in 2005, $2025. But the thing that I want to say here in this talk is that it's really, for all the papers that have where we've described and published bar tools, the important thing is the talking to all of the co-authors and to develop these tools, dozens and dozens of emails to get the visualization right and to make that view available to the community properly. Those are the papers where Arabidopsis data sets or tools were released. As Siobhan mentioned, I've also been involved in the Multinational Arabidopsis Steering Committee and something called the International Arabidopsis Informatics Consortium, which arose as the NSF withdrew funding from TARE in the early 2000s, mid 2000s. And we were trying to set up something called Airport to sort of help the Arabidopsis community. And then at the same time, TARE actually continued on to become a not-for-profit corporation. NSF, in its wisdom, withdrew funding from Airport. And then there was an elaborate rescue operation to take parts of Airport and distribute them among TARE and CGR and the BAR. And you can read about that in the plant cell paper. I'm also involved in something called the Plant Cell Atlas, where we're trying to set up an infrastructure for single cell plant data, which hopefully will end up in the Single Cell Expression Atlas at the EBI through community curation effort. And also, as Siobhan mentioned, I have been sort of a stealth Arabidopsis promoter in many of the courses that I've put together on Coursera, not only plant bioinformatics, but also bioinformatic methods 1 and bioinformatic methods 2. And these have been quite well received by the community. All right, with that, I would like to thank all of the students and to you and to the organizers, to NAASC, for this award. And thanks to you for listening.
Thank you very much, Nick. Our last introducer will be Marcella.
So I am delighted to introduce today Dr. Anna Stepanova. Her research is centered around plant hormones and synthetic biology. For today, we're celebrating her outstanding contributions to dissemination of Arabidopsis. She has really contributed to enhancing accessibility, communication, and integration of Arabidopsis knowledge. And she has really impacted researchers, educators, students, and a lot of young people. So for those who know Anna, you would not be surprised that she excels at all 3 different categories for this award. So she has committed an incredible amount of time developing teaching and outreach tools that use Arabidopsis as key components of her modules. She has worked with children and teachers, and she will probably talk to you about more details about her Plant for Kids program that has impacted or provided opportunities for students in the department outside of her lab for outreach activities and leadership programs. She also developed courses that use Arabidopsis as tools for molecular genetics. And finally, she has also contributed several review articles that highlight the role and the impact of Arabidopsis research on crop studies. So these are really, really important documents or white papers that can be used for justification for funding for Arabidopsis, which is really impactful for our community. So I want to congratulate Anna on this well-deserved award, and I look forward to your talk, Anna.
All right. I'm sharing my screen. All right. Thanks, Marcella. And I'm very honored and humbled to have received this award from the Arabidopsis community. I would like to talk about my plant knowledge dissemination efforts that have taken different shapes and forms, including that in teaching, community service, science communication, and public outreach. So I thought I'd take my 6 minutes to share some of that experience and hopefully encourage others on the call to get involved in similar efforts as well. Many of us on this call are faculty, students, and postdocs, which means we get to teach. And the courses that we are assigned to teach may not have a formal plant component, which is the case for my courses as well. I teach an advanced molecular genetics course to about 30 graduate students every fall, and I make a deliberate effort to use some plant examples in my lectures, such as this very visual case of transcription factor mutants with really cool flower phenotypes that I like to show. And in the hands-on synthetic biology lab course, I intentionally included a plant biosensor module where students got to build an ethylene transcriptional reporter and worked with tiny Arabidopsis transgenic seedlings, as you can see in this slide, to visualize the biosensor activity using microscopy. And for most students, many of whom are chemical engineers, microbiologists, animal scientists, this is the only exposure to plants in their entire careers. So I do my very best to make their experience with plants engaging and memorable for all students. Besides teaching specific classes, faculty, students, and postdocs often get asked to give a guest lecture, for example, to replace a colleague that is traveling or has fallen ill. So I encourage you to consider having a slide deck ready that features plants. I have two go-to lectures in those cases, one specifically on Arabidopsis as a model system that I prepared for an undergraduate level audience, and another is an outreach lecture for high school students and above on heirlooms, hybrids, and GMOs. The second topic is of interest to a variety of audiences, especially among gardening fans, and has been a big hit. Another way to deliver your message is through formal publications in scientific journals. Of course, the caveat of this approach is that review and opinion articles and white papers that we write are usually primarily read only by scientists and really rarely make it into the real world to reach the general audience. So if our papers do not reach the public, then we go to the public ourselves. My lab members and I developed a week-long teaching module for high schools on recombinant DNA technologies, and we beta tested our experimental pipelines, our protocols, in the hands of 43 advanced placement biology students at a local high school, Athens Drive. We then, for 2 summers, ran a 30-hour-long training workshop for high school teachers. And with the 23 teachers trained to date in recombinant technologies, we enabled them then to borrow our miniature lab equipment from our mini library so that the teachers can implement the hands-on modules that we developed in their classrooms. And several of the teachers that we've trained took advantage of that opportunity and were able to introduce their students to concepts of basic cloning and recombinant DNA. And for younger audiences, for the past 15 years, my group has been running a Plants for Kids program at the North Carolina Museum of Natural Sciences. We developed our experimental modules specifically for elementary school children, so they're adapted to their young minds. And to date, we carried over 130 live demos, ranging in duration from 2 hours to 8 hours each. We have also made a number of classroom visits with our Plants for Kids program and ran demos at science fairs and other school community events. And to further broaden the reach, for the past 10 years, I have worked with graduate students taking my molecular genetics course to make educational videos for extra credit. This is a very popular option with students. This course is taken by about 30 students every fall, and typically about a third of them commit to working with me throughout the semester to make short animated videos about the topics students are learning in class. The goal here is to explain a complicated concept, often an advanced experimental method, at a level accessible to the general audiences. That is, to folks with a high school level of understanding science. And some of the videos that my students have made accumulated several thousands of views. I think the record is like 110,000. So their video projects are getting noticed and helping folks learn. And all of the outreach resources that we developed, including lecture slides, protocols, and the videos, are freely available through our lab's webpage and through YouTube. And a more formal way to make a difference and get involved with the Arabidopsis community is to serve in the leadership of our grassroots organizations, such as North American Arabidopsis Steering Committee or Multinational Arabidopsis Steering Committee, as I have done since 2018. And if you are unable to commit to a leadership position, please make an effort to participate in various events and initiatives that these amazing groups organize. And this wraps up my very short talk. I'd like to thank my past and present lab members, undergraduates, visiting scientists, collaborators, colleagues, and mentors who have all participated in my group's outreach program over the years. It takes a village to get our message across that Arabidopsis, genetics, and plant biology are all exciting and worth studying. And thank you for your attention.
So now we're going to move to our question and answer session. And so if folks want to ask a question, they can raise their hand or put it in the chat. And while people are thinking of questions, I want to ask one. And so it's been interesting listening to you all talk. I've used data, transcriptomic data generated by all of you or all of your labs. And so it's fun to see this all come together. But I've utilized that data through Nick's website. And so I wanted to ask Nick a question about this tremendous resource. The statistics you shared of how much this is used are not surprising for those of us who use it routinely. But this is a challenge for maintaining this, adding this. And so can you tell us how you make this happen, how you support this, and how we as a community can do more to help you get the support you need to generate or share the data that we all use?
Right. Well, so funding is always an issue, that's for sure. And I've been fortunate to be able to secure grants over the past 15 years that have helped me pay for a bioinformatic technician to maintain the bar. But I'm also very privileged to be at the University of Toronto, which is the best school in Canada. And we have access to many great students, including undergraduate students who actually pay to take a course with me and who develop modules or process data or do analyses that support the bar. So in a way, it's kind of a funny setup in the sense that we can leverage students, and they oftentimes get their names on papers too. So it's like a win-win for everyone, and that's how we can that's how we've been able to keep things going for so many years.
Well, it's been fantastic. We appreciate it. So are there questions? So while others are thinking them, to any of you that choose to answer, we're really trying to understand what advice you might give to young investigators who want to go down these paths, explore things. What would you recommend to them to enable a path where they think not just about their own research and their own needs, but also benefit the larger community through sharing data or other ways?
I have a short answer that I can give. One, there is no good time. So like, don't keep thinking, oh, when I'm tenured, I can do this and that, or there's no good time. It's something that once you do it, you'll see you can do both. And second, I will recommend finding something that you authentically and genuinely feel like you can contribute in. So something that you feel that you have an added benefit in and you're genuinely interested in helping. Otherwise, from my experience, it's hard. It does take time and it does take the effort. So you need to really want to do it and think you're important in doing that. So yes, those would be the two things. Just get started and get started with something you really believe in.
Others want to answer that?
I would second this, and I would also suggest that you do what feels right for you. When I had my biological kids, my own children, I would be spending time with them to entertain them over a spring break, for example. So that's how Plants for Kids was born. We started doing fun experiments at home, and then the friends of my kids joined, and then somebody else said, why don't you share it with the rest of us? We want to know what you guys came up with. So I developed, together with my husband, this Plants for Kids program, starting with my own family, and then knowing that it works in the hands of my children, made it available to others. So as my kids grew, so did their interests. So our initiative to do high school initiative things became a reality once my kids were in high school, and I made those connections with high school teachers. So as you're making those connections with the right audiences, you develop the activities that fit that audience level-wise, intensity-wise. And right now I work with graduate students because this is what I have the most exposure to. But I know the level of understanding and thinking by younger children versus middle schoolers versus high schoolers. So this personal experiences help kind of shape the types of activities that my group develops.
Thank you, Anna. Others?
Yeah, and I can also add to that. I could say a lot of the web applications or the information that we've developed or tried to share stem from frustration, right? I mean, and I think that that's a place where you sit there and you're like, you want things to be, you have so many other things that are difficult, including, doing these experiments. So you want to make things easy, and you try to find a way. And if the time when you're doing the actual data analysis or searching through multiple papers trying to find information, if that's the time, right? That's the time when you think about how can I make this easier and more accessible, right? Or at least your work more easy and accessible to the community. So I would say, yeah, think about the things that frustrates you, right? And think about how can you improve this by sharing in the community. That's how I see it, yeah.
And maybe just to add also the importance of communication and like talking with people. I feel like the Arabidopsis news group was really something that I really profited from as a starting out researcher. But there are other channels these days, and just kind of take advantage of those to use that as a sounding board, or to ask people, what's available. Could I make something like this? Does it already exist?
Other questions? I have another one, unless somebody else has one. And so, you've all talked about how to get started. And so what inspired you to start, and what feedback have you gotten from the community that keeps you going? So you can pick on either or both of those.
Well, maybe I'll go first. So I just the confluence of kind of the internet and big data was really sort of an eye-opener for me, and I really wanted to make the data accessible to the community through the internet. So that was kind of the inspiration for the Botany Array resource, or the BAR, and then also for to create a cool visualization. It was just so much fun. And maybe to touch on what Dawn said, the frustration of not being able to access data easily, and just being inspired by Sean Cutler, his GFP fusion screen. It was kind of, okay, can we do this? Can we actually make the data accessible in this visual way? And when I first saw it, it was like, wow, this is really awesome. And it was the feedback that I've received over the years on the EFP browser has been phenomenal. And I remember perhaps a highlight for me was having a student I think in Finland, wanting to take a selfie with me as the creator of the EFP browser. And I'm like, okay, I've made it. So, yeah.
That's great. Thanks. Others?
For me, since my main program is the Plants for Kids, it's the children that come repetitively, more than once, basically, to the museum just to share in their excitement how their plants grew and the fun things that they have observed. It's the excitement in the eyes of the kids and their little stories. And some of them would bring us postcards that they drew to thank us for the ideas that they gave them. And the parents are very grateful as well, because their kids were busy over a spring break or over a summer break, as opposed to just sitting in their iPads and doing electronics all day.
I will just reiterate what Nick said and also Anna said, right? I think it's just getting feedback from people who have used, the resources or the information that you've shared, and just hearing from them how it has helped them, to with their research or with sharing information. A lot of undergraduate students actually use our web browser, and that has been really rewarding. And quite a number of students I've run into at meetings where they're like, oh, we've used your browser to look at this or that. And I think that that's really, it makes it worth it, right? Because you know that it's being used by the community. So.
Oh, yeah.
Yes, please.
Yeah. Well, first for Anna, and then sort of a similar for Nella. But first for Anna. So you've done for so many years. You've done so many programs, a lot facing the public at various ages, and I guess probably some of them were funded by grants, because I'm not sure. I guess my question is, how would you as a faculty member find time to sort of do these, when it doesn't seem to me something that maybe that your administration would count as part of your normal work. I mean, the lectures in class, perhaps. So is the things that you're doing to the public, is that additional, or is it somehow count as part of your regular workload? I'm wondering how other faculty might incorporate some of this.
Well, we each have a service component to our appointments. For some, it's 5%, for some, it's 10%. And then we're basically given the freedom, whether you're serving on university level committees or society committees, or you're going out to a classroom and doing, I don't know, reading to kids. It's up to you how you put that service into practice. I've done a bit of both, like at the university level and outside of the university, and my own interests evolve. But if you're doing something very substantial, like a teacher training workshop, you need to have dedicated funding because you need to pay the teachers to come. And the reason why is because they're taking their personal time off during the summer, and then they're spending that time with us. So you need to have them, you need to give them a little stipend, and you need to have materials and supplies that also cost money. But for things that we do at the museum, we use all recycled materials, like recycled yogurt cups, autoclaved soil that we no longer use, even napkins or shredded junk mail can be used as a substrate to germinate seeds. And as a seed source, we buy a 1-pound bag of peas or beans, and it lasts us for a couple of years. And it's not seed grade seeds, but rather food grade seeds. And they work wonderful as long as they're dry and uncooked. So a lot of things can be done with very little money or completely for free, just being creative.
Thanks. Thank you, Anna. And then my question similarly for Nadanella was, I was wondering if you, in your experience still being like in the lab all the time, is there like a low activation energy effort or something that you could see for either undergrads or grad students that where they could get involved? Like maybe you give talks on podcasts. So, you know, what is something around you that you think that people could do pretty easily? Maybe a podcast or a blog or something?
First of all, yes, those are amazing suggestions that you just gave. And I think those are great opportunities. But even thinking of what I do in classes, you know, I wrote the book, but anyone could actually use that book to get into classes all over the US and the world, because it's just a children's book. It's a story, and it entails within it like parts of plant biology, like the difference between monocots and dicots, the meristem, and all kinds of like very basic but real plant biology. So what I usually do, I give the teachers ahead of time a couple of books. They read it to the class, and then I come in and we do this discussion and we talk about it. So that's, for example, something very simple that anyone could do. And from my experience, it's really fun, both for me. I always get into class with a bit of hesitation, you know, they might not be interested at all. Not everybody loves science at that age or at all. And every single time, not one time I left a class and was like, they didn't get it. They are so engaged. And it's for me, it's uplifting to get out of the class every time, because again, that's something that I really love. I really love plant biology. I really love what I do. And just sharing that and like sharing the excitement like both ways is just really fun. And I think super easy. It does take time. It's true. There's nothing to say about it. It's extra time that you spend on doing so, but I think the reward is much higher than the time you actually give, and just the reward of the feeling you get by doing that.
So I had a follow-up question. Anna mentioned NSF broader impacts, I think, and how that supported activities and motivated many of us to develop activities and to reach out to a broad, diverse population of researchers. And so recent changes in the way the priorities of the National Science Foundation are affecting what we do. And so I don't know if anybody wants to think about other ways to how these changes are impacting broader impacts, or other ways that we can motivate ourselves and others to really engage in these through a reward system like funding or other ways.
I had the discussion with an NSF program officer about that, and they've said specifically that the broader impacts are still a thing. The only difference these days, instead of saying that we are developing this particular activity for a specific group in mind, you have to say, well, we're developing it so that it's accessible to all. If in the past we used to highlight that, well, our Plants for Kids activities are bilingual, English and Spanish. Now we just say they're accessible to all. We don't emphasize the word Hispanic. We don't say it's only for Spanish-speaking people. We say these activities are accessible to all. And that meets all the expectations of the NSF, and they're very happy with our broad impacts.
Well, it seems like we then have reached a great ending point with 1 minute before the hour. So thank you all very much to the awardees for all the work that you've done, for those of you who've introduced them, and for those of you who've taken the time to participate today or watch this video. We appreciate the ways that you all have built the community. Thank you very much.